pleasure to be here with you all today to talk to you about my project on DNA nanotechnology, which I did under the mentorship of Dr. Rafael Carrascosa-Marzo. So, in this project, I explored how DNA nanostructures, specifically a tetrahedron, can be used to deliver chemotherapeutic drugs directly and more precisely to lung cancer cells. So my goal was to address some of the key limitations that we see in conventional treatment methods like chemotherapy today, and use DNA as a targeted delivery system. And this focus naturally led me to one of the most persistent and challenging areas around medicine, cancer. Firstly, I'd like to talk a bit more about the background of cancer and what inspired me to pursue this disease for my project. So, cancer is one of the oldest and undoubtedly among the most studied diseases in medicine. However, despite decades of research, it remains one of the leading causes of death annually. In fact, after cardiovascular disease, it is the second leading cause of death globally. In each year, over 10 million people die from cancer-related illness, and whilst we've made significant progress in screening, diagnosis, and therapy, certain types of cancer remain particularly difficult to treat. And among these is lung cancer. It is the most commonly diagnosed form of cancer worldwide, and unfortunately also one of the deadliest. Amongst its forms, non-small cell lung cancer, which I will be referring to as NSCLC, is by far the most common, and it makes up over 80% of all cases. And what makes NSCLC especially concerning is its survival outlook. Compared to other major cancers, this has the lowest 5-year survival rate, averaging just 21% across all stages. And unfortunately, this figure has not improved substantially over the recent years, and many patients are still diagnosed at a late stage where curative options like chemotherapy and surgery are no longer possible. And even when chemotherapy is used, relapse and treatment failure are unfortunately still common. So, this disconnect between how widespread NSCLC is and how limited our treatment outcomes are raises an important question for me. Is there a way to make chemotherapy more selective, more controlled, and most importantly, more effective? So, this is the question that led me to explore a growing area of research that uses DNA in an entirely new context. Instead of focusing on its biological role, scientists have now begun to use DNA as a structural and programmable material. So for anyone unfamiliar, DNA is a biological molecule that carries our genetic information, and it's made up of 4 basic units, A, T, G, and C, that pair up in a predictable way. And scientists can use this predictability to design short DNA strands that stick together in specific ways, almost like building blocks. By designing these short DNA strands, researchers can build nanoscale structures that fold into 3D shapes and carry drugs like cisplatin, which is a chemotherapeutic drug, directly to tumor cells. As you can see in the figure above, the 4 strands of DNA can self-assemble into the tetrahedral shapes, as you can see. And what's great about these structures is that they're stable, customizable, and can release their contents under the conditions they're programmed to release them in, such as inside a cancer cell. However, before turning to newer strategies, it was first important for me to understand where current therapies like chemotherapy still fell short. So, chemotherapy, as we all know, remains one of the most widely used tools in cancer treatment. Drugs like cisplatin, which is the most commonly used drug to treat non-small cell lung cancer, work by damaging the DNA of rapidly dividing cells, preventing tumor growth. However, this mechanism is not unique to cancer cells. As we can expect, many healthy cells in the body also divide quickly, and chemotherapy cannot distinguish between these healthy cells and cancerous cells, and therefore affect these healthy cells as well. And this is the reason why most chemotherapeutic drugs are associated with side effects, including but not limited to nausea, fatigue, immune suppression, and organ toxicity. This means that even when treatment is effective, it comes at a considerable cost to the patient's well-being, which we do not want. Another major problem is drug resistance. Tumor cells can actually evolve ways to pump drugs out of cells, repair their own DNA damage, or reduce the drug uptake entirely. As you can imagine, this makes treatment far less effective over time, even after an initial positive response. Finally, chemotherapeutic drugs circulate systemically throughout the body. They don't naturally concentrate at the tumor site, which means it's difficult to achieve high enough local concentrations where the drug is wanted without affecting the rest of the body. So, recognizing these challenges, researchers have turned to more targeted drug delivery systems in an effort to reduce toxicity and improve treatment outcomes for patients. One particularly promising direction is the use of DNA nanostructures, like I mentioned, an idea that offers several advantages over conventional drug delivery. So why DNA nanostructures? These structures can be engineered to improve the precision of chemotherapy, directing drugs to cancer cells while limiting exposure to healthy tissue. Secondly, because they are made out of DNA, which is not foreign to the body, they are typically biocompatible, which means that they do not trigger an immune response, unlike synthetic nanoparticles. Also, DNA nanostructures can help bypass drug resistance mechanisms by increasing the intracellular drug concentrations and avoiding some of the pathways that cancer cells use to evade treatment. And this means that low dosages of the drug are sufficient to kill cancer cells. So after covering the basics of the problem and a potential solution, I'd now like to talk about my methodology that I followed to design my own nanostructure. So my aim was to assess whether the structure could remain stable, selectively target cancer cells, and support a more controlled release of the drug. To do this, I worked with various computational tools to design, assemble, and simulate the environment that the nanostructure would be in, and evaluate how it might behave in a biologically relevant environment. So the first step in my project was to find an optimal sequence of the bases, A, T, G, and C, to make the strands out of. Then, using these generated sequences, I basically built the tetrahedral shape on another software called OxView. Then, after checking that the structure was in fact stable, I worked on a viable targeting mechanism that would, in a way, instruct the structure to travel to cancer cells to deliver the drug. And finally, I ran some simulations to check that the structure remained stable in biological conditions. So on the screen, you can see a table and two key diagrams that I use in the design process. The table on top shows the bases on the 4 strands of DNA that I used, and the matching colors show the complementary strands of DNA, which make up each side of the tetrahedron. So the 6 different shapes make the 6 different sides of the tetrahedron. So the way I obtained these sequences of bases was using the software NuPack, which showed me the most thermodynamically stable sequence of bases that would resist degradation in the human body. On the bottom left, figure 1a, is a diagram showing the layouts of the strands before self-assembly. And on the right, 1b, is a net diagram, which is basically a flattened view of the tetrahedron, which is similar to how you might look if you unfolded it and laid it flat on a piece of paper. Then, I added intentional breaks in the DNA backbone, called nicks, to improve the flexibility, which are indicated by the arrows as seen in the diagram. I ran 10 different sequence trials and selected the one that showed the strongest predicted stability for the next step. So once I had finalized the base sequences and layout using NuPack and OxView, the next step was to build and visualize a 3D structure. On this slide, on the left, you can see the 3D model of the assembled tetrahedron, which I created using the software OxView. Each DNA strand was positioned to match the intended folding from the earlier 2D design in the last slide, and I also included the same nicks introduced in the design stage, so the model would accurately reflect how the structure would fold and behave in a real biological setting. And the gray mass you can see in figure 2a is actually the targeting mechanism used, which I will talk about later on. Then, I ran a relaxation simulation to let the structure settle into its most stable form. To test its stability, I performed a 10,000 step simulation, which means 10,000 frames, that tracks how the structure behaves over time by tracking the movement of atoms, to see if the structure holds or begins to unravel. So as shown in figure 2b, the tetrahedron did hold together well. So some minor movements were seen, but the structure remained intact. And this suggested that the structure could survive long enough in the human body to reach its target. And figure 2c, finally, shows a degraded tetrahedron, which is what we would expect to see to happen in the tumor cells in order to release the drug that is inside. So after confirming that the structure was stable, the next step was to load it with cisplatin, the chemotherapeutic drug, and guide it to the right target, the cancer cells. So the actual process of attaching the drug was relatively straightforward. Cisplatin naturally binds to guanine bases, one of the 4 bases in DNA, so the strands were designed to include binding sites that would allow the drug to attach covalently during self-assembly. Now, the most tricky part was not loading the drug, but it was making sure it reached the right cells. Like I said, since the drug also affects healthy and cancer cells alike, I needed to find a way to direct the nanostructure specifically to tumor cells. And this process was definitely the most challenging and took a lot of trial and error. But the most viable idea I settled on was a well-known feature of non-small cell lung cancer cells, which is the overexpression of EGFR, or epidermal growth factor receptor. This is basically a protein that appears in much higher amounts on cancer cells compared to healthy ones. So to use this feature, I used cetuximab, which a monoclonal antibody, meaning the shape is complementary to the proteins on the cancer cells, that binds tightly and specifically to EGFR. By attaching cetuximab to the vertices of the tetrahedron, I gave the nanostructure the ability to recognize and attach to the NSCLC cells that express these high levels of this protein. So once the structure binds to the EGFR on the cancer cell surface, it's taken into the cell through receptor-mediated endocytosis. And this is basically where the cancer cell almost engulfs the structure. And inside the cell, the conditions are more acidic due to the endosomes and lysosomes. And this drop in pH is what causes the structure to break down and release the drug directly inside the cells. So this targeted release has two advantages. It helps overcome drug resistance, and it reduces off-target toxicity, since healthy cells are less likely to take in the structure. Another benefit of this system is that the breakdown products, which are the fragments of DNA left, are non-toxic and can be easily broken down by the body and cleared. So this helps reduce the long-term side effects that I previously mentioned, or unwanted accumulation in tissues. So, while the simulations and design outcomes from this project were promising, there are still several key limitations to consider. Firstly, everything I've shown so far is based on computational modeling, or in silico. While simulation tools are valuable for predicting how the structure might act, they cannot fully replicate the complexity of a living system. And even when we experiment on tissue cultures, we still don't know for certain how the entire body might react in a real life. For example, we might see success in tissue and lung tissue cultures, but another organ or system might be affected by the drug that we do not know of until we can test in vivo, or in real living organisms. So for this approach to move forward, it would first need to be validated experimentally and eventually in clinical studies to confirm safety, targeting accuracy, and therapeutic benefits. Although some early-stage experiments have been done on small animals like mice, we don't know how these DNA nanostructures might act in human bodies, where we have a different metabolism and different physiology. Another key limitation is the pH-targeted release mechanism. So while tumors are acidic and would cause the structure to break down, there are sites like inflammation or immune cells that also have a low pH. This raises the risk of premature drug release in areas where we do not want the drug, like in healthy cells. To improve the specificity, future designs could incorporate a dual trigger system, almost like two-factor authentication, such as enzyme-sensitive bonds to control where and when the drug is released. However, despite these gaps, the potential still does remain strong. With continued research and refinement, DNA nanostructures could help address limitations in today's cancer treatments. And looking ahead, the potential of DNA nanostructures in cancer therapy is definitely promising. With relatively small adjustments to targeting mechanisms and release conditions, this approach could be adapted not only to NSCLC cells, but in principle to other forms of cancer as well. While this project focused on this specific system, the broader platform is flexible and highly customizable. So future research involving especially in vivo studies and clinical translation will be essential to understand how these structures behave in a more biologically relevant environment. But with continued development, this technology could move us to closer to more precise and less toxic cancer treatments that are tailored to the needs of each patient. So, in conclusion, DNA nanostructures offer a promising strategy for making chemotherapy more targeted and precise. In this project, I explored a viable design for delivering cisplatin, the chemotherapeutic drug, directly to NSCLC cells, using a tetrahedral DNA structure. While this does remain at an early stage, the platform shows strong potential, and with further development, it could be extended beyond NSCLC to other cancer types as well. Thank you for listening. I'd be happy to take any questions.
Thank you so much. Excellent presentation. Should we have any questions? Please go ahead.
First of all, really good job. I love the presentation. It was super clear. I would say, do you have any like future steps for your project specifically? Like, what do you plan on doing?
Yeah, so I think, like I mentioned before, although this project was definitely really interesting, the main limitation I faced was not having access to an actual wet lab and not being able to experiment on, for example, small animals or maybe even tissue cultures, since everything I did was basically software. So I think if I had the chance to actually work in a lab environment where I could see those changes, especially using, like, for example, scientists use fluorescent markers to see how the drug is taken up, I think that would be my next steps in this project, to actually see how it works in a real biological system.
I think you also mentioned about dual trigger systems and
Yes.
other future works. That sounds exciting as well. Any more questions? Please go ahead.
I'm not too sure on the topic, but wouldn't DNA get broken down in the cytoplasm if it was recognized?
Yeah, that's actually a great question. That's one of the problems that we're facing as well. I think the main thing that stops the DNA from being broken down is the fact that it is synthesized DNA, and also it's attached to those, it has a EGFR that brings it to the cancer cells. So I guess the longer it would remain in the human body, the more likely it'd be to be broken down before it reaches the target. But obviously it depends from cancer type to cancer type, but for example, when you're targeting lung cancers, you have to keep the drug, you have to keep the structure in the body for the shortest time possible for it to not break down. But I think that is really a good point, and one of the another key limitation in this project as well.
Probably that's where the triggering mechanism helps, like the pH level
Yeah,
you mentioned.
definitely, yes.
You
Yeah.
need to keep it encapsulated until it reaches the
Yes, exactly.
That's a really good question. I think it kind of adds up in a similar topic. We heard about AlphaFold, so it's one step higher. Probably can I ask about the time it takes for you to run OxView, which is a thermodynamic stabilization software?
So the number of steps I chose, which is the benchmark for this type of field, 10,000 steps, it really didn't take very long, because I think partly has to do with how small the structure was. So it was 20 base pairs on each side of the tetrahedron, so it doesn't take more than 10 minutes, which I think is important. But again, with bigger structures, it would take a lot longer, but thankfully in my situation it didn't take very long.
A cool perspective to look at is the 10,000 frames is not the right way. I think it's a different scale.
Yeah.
Do you know nanoseconds or femtoseconds being used on 10,000 frames could be any
Yeah, that might be the
seconds.
case actually,
could
yes.
be the
Yes.
10 nanoseconds.
Yes.
Probably if it kind of scaled up, it lies between AlphaFold and pure quantum chemistry studies, so it's a really good way of doing this. Thank you so much. Let's thank the speaker again for an excellent presentation.